Hello, my name is David Bressler, and I am presenting Figure 5 from the paper Unraveling the Mechanisms Involved in Motor Neuron Degeneration in ALS, written by Lucy I. Brujan, Timothy M. Miller, and Don W. Cleveland. Amyotrophic lateral sclerosis, or ALS, is the most common adult motor neuron disease. ALS is responsible for the dysfunction and death of motor neurons. This leads to many symptoms such as muscle atrophy, generalized weakness, overactive flexing, paralysis, and then ultimately death. Fatalities caused by ALS are generally due to failure of the respiratory muscles, which usually occurs between 1 and 5 years from the onset of symptoms. The direct cause of ALS is not completely known. However, there are many different pathways that each affect neurodegeneracy. Some of these include problems in the SOD1 gene and growth factors. By studying these different pathways, a treatment may be developed that can help with slowing or even stopping the onset. The SOD1 gene is responsible for making an enzyme called superoxide dismutase. This enzyme binds to copper and zinc and breaks down superoxide radicals, which are charged oxygen molecules. These toxic molecules are byproducts from normal cell function that, without being removed, will cause cell damage. Geneticists have found a mutation in the SOD1 gene that could be responsible for cell degeneration. The authors found different mouse models with a single amino acid change in different spots. These mutations include SOD1 glycine at position 85 to arginine, SOD1 glycine at position 37 to arginine, and SOD1 glycine at position 93 to alanine. All of these models showed that weakness developed in the hind limbs, which coincides with motor neuron degradation. Neurotrophic factors, usually peptides and small proteins, support the growth and development of neurons. A neurotrophic factor that may be involved in ALS is insulin-like neurotrophic factor 1, or IGF-1. IGF-1 is a hormone used for cell growth by almost every cell in the body, especially nerve cells. Figure 5 shows insulin-like growth factor 1 was delivered directly to the neurons using an adeno-associated virus vector, or AAV. This vector is used because it has low immune response, so the immune system will not try to destroy it. AAV is also used because it can incorporate the desired gene into the genome of the cell. The AAV vector with IGF-1 was injected into the hind leg of the SOD1 G93A model. The IGF-1 injection seemed to be effective as it prolonged the lifespan. However, it is uncertain whether the IGF-1 had any effect on the neurons which already produce their own hormone. It is possible that the added IGF-1 may have upregulated the production of IGF-1 in the cell and this caused the observed change. The authors are also unsure of whether neighboring neurons or astrocytes are affected by the IGF-1. These results show that adding additional IGF-1 directly to the affected cells is beneficial to the host. However, it is unknown which pathway is directly responsible for the positive outcome. The authors have concluded that there are many pathways that are potentially responsible for causing ALS, so being able to find an effective treatment is very difficult. This can be seen as adding IGF-1 does not stop the progression of ALS, so this is not a sole cure. This paper does demonstrate that these pathways can affect not only ALS, but may also affect other neurodegenerative diseases such as Huntington's disease. The research involved does not only provide a stepping stone for ALS treatments, but any other neurodegenerative disease as well, because they may share many similar pathways. Future involvement could include searching for similar pathways in other neurodegenerative diseases to try and find similarities which can ultimately result in possible treatments. Thank you.